Good morning, I'm Nell Muldoon, and here's the latest update from Spectrum Autism Research. Autism-linked protein screen reveals hundreds of new interactions by Grace Huckins. A new analysis has uncovered more than 1,200 new protein-protein interactions involving the proteins encoded by 92 genes strongly linked to autism, according to unpublished results. Researchers presented the findings virtually at the 2021 International Society for Autism Research annual meeting. Though scientists have extensively studied many of the autism-linked genes considered in the new work, including SHANK3, CHD8, and FOXP2, understanding how their protein products interact with other proteins in the cell could provide new clues to those genes' functions, says study investigator Snow Nang, a graduate student in Nevin Krogan's lab at the University of California, San Francisco, who presented the findings. It could also reveal how mutations in those genes contribute to autism. Genes are great as an initial point, but at the end of the day, proteins are the ones that carry out a lot of the functions, Nang says. And proteins, they never really function on their own. They always interact with other proteins. Previous studies have cataloged numerous interactions involving autism-linked proteins, which are available in online databases such as the Human Reference Interactome. But, Nang says, the results of protein-protein interaction assays can vary a great deal depending on the methods used. The data in the Human Reference Interactome come from experiments in yeast, for example, but Nang and her team did their experiments in human cells. Probing proteins. The researchers attached each of the 92 autism-linked proteins to a protein complex, which they used as a tether to pull the protein out of the cell, along with any other proteins it interacted with. They identified those other proteins using mass spectrometry. After factoring out the background noise of proteins that just happened to be floating around nearby, Nang uncovered 1,403 protein interactions involving the autism-linked proteins. 87% of those interactions are not listed in the Human Reference Interactome or other similar databases. She and her team are studying how mutations seen in autistic people disrupt these interactions. The analysis relied on human kidney cells as the first test because they're easy to manipulate, Nang says. But kidney cells and neurons express many distinct proteins that allow them to carry out their particular functions. For this reason, Nang and her colleagues next plan to run the same experiment in neural progenitor cells, the type of cells that ultimately develop into neurons. We of course expect to see differences because different cell types will have different interactions, she says, but we also expect to see a lot of intersection. The post Autism-linked protein screen reveals hundreds of new interactions, appeared first on Spectrum Autism Research News. That's all for today. Check back next Monday for more content from Spectrum Autism Research or go to spectrumnews.org.